Hello, I hope everyone can hear us now. The WebEx says that we have audio, so hopefully it does. My name is Jason Church. I'm with the National Center for Preservation Technology and Training. We are research office of the National Park Service. And I'm here today to introduce our first, what will be a series of conservation science webinars. And the first is Scientific Method and Experimental Design in Preservation and Conservation Research. And for this, I would like to introduce Dr. Chandra Reedy, conservation scientist and professor at the University of Delaware, and she'll be doing this webinar today, the lecture today. Will be in 3 parts. At the end of each part, we'll take time to have about 10 minutes worth of questions. We do not have audio for the participants, but you can chat in your questions, and then Dr. Reedy will pull up those questions. Also, we have handouts for both if you want to take notes on the PowerPoints and also experimental exercises that Dr. Reedy will be doing later. And those can be found on the NCPT website at ncptt.nps.gov/blog/scientific method and experimental design. Or you can go to our homepage and there's a link right there on it. Download those PDFs, and that is also the link where we will place the recorded lecture from today when after it's been processed. So without further ado, I'd like to turn it over to Dr. Reed.
Thank you, Jason, and hello everybody. Welcome to this webinar. First, I should introduce myself a bit and explain why I'm so interested in scientific methodology and experimental design issues. I'm Chandra Reedy, and I originally did my PhD work at UCLA in a program that was interdepartmental, and it required work in at least 3 different departments. So I ended up being 1/3 Earth and Space Sciences, 1/3 Anthropology, and 1/3 Art History. But my dissertation research was actually mostly carried out in the Conservation Center of the Los Angeles County Museum of Art, where my primary advisor was located and where I ended up working for the first few years after getting my PhD. So I've been working in a very multidisciplinary way that incorporates natural science, social science, and humanities research along with heritage conservation science. And this really requires that you think carefully and be clear about your experimental design. I've been at the University of Delaware since 1989, where I've taught in the graduate program in conservation, the PhD program in art conservation research, the museum studies program, and in the graduate program in historic preservation. I'm currently in the Center for Historic Architecture and Design, where I also serve as director for its Laboratory for Analysis of Cultural Materials. So I still incorporate geology, anthropology, technical art history, heritage conservation science, and historic preservation research into my research program, which means I'm constantly having to think carefully about my research designs and experimental design procedures. Early in my career, I co-authored books on experimental design and statistical analysis in conservation research, and this required first reviewing 320 research papers to examine and try to take apart the experimental design structures of each of those. And I found that very helpful, and I've tried to use that information over the years to help students on their experimental designs and to try to polish up my own research designs. I've continued to pay careful attention to research design while I served for 7 years as editor-in-chief of the Journal of the American Institute for Conservation, and for the last 6 years as editor-in-chief of Studies in Conservation for the International Institute for Conservation of Historic and Artistic Works. That requires reading through a lot of papers, many of them outside of my area of expertise, but trying to look carefully at experimental design issues. I've taught a workshop on this topic many times at many venues, and now this webinar can be made permanently available so anyone can access it at any time and from any location thanks to NCPTT. So I'd like to first go over some of the main goals of this webinar. First, we'll be reviewing what I think are the most crucial aspects to consider in experimental design. We won't be talking about everything having to do with this subject. It's a very, very large subject. We'll be discussing experimental design and not statistical analysis, although, as we'll see, a valid statistical analysis does require a proper experimental design, so they certainly are related. And third, many of you, perhaps most, if not all, will have heard some or even all of these ideas before, maybe multiple times, certainly, I hope, in science coursework in school. But I myself find it helpful to review and rethink about these issues from time to time, to renew and refresh my ability to organize research. So I think review of these concepts is always helpful, at least it is for me. And fourth, today we want to think about how these experimental design concepts can be used in daily work for improvements in clarity of our thought processes, improvement of design, of testing procedures, and perhaps lead to outcomes in which you have more confidence. In preservation and conservation, we constantly have to make decisions that it can have a major impact on culturally significant objects and important irreplaceable collections. We have to decide what preservation measures and conditions to choose, what treatments to choose, how to carry it out. Is one choice really better than other possible choices? So the purpose of good research and experimental design is simply to help us make the best possible choices and to have confidence in that choice. And finally, a better understanding of what makes for good experimental and research design can improve our ability to evaluate the professional literature and as well as improve our ability to contribute to that literature. All of the PowerPoints that you see today are available to download on the NCPTT website, either to jog your memory later or to print out and take notes as we go along. References for all the papers that I mentioned today are also given there, as well as some practice exercises. The webinar is structured into 4 parts. Part 1 will review the scientific method in general. We'll discuss first a seminal paper by John Platt about what he thinks accounts for progress in scientific research. Then we'll look at what Enrico Fermi and Louis Pasteur have had to say about the process of thinking in science and how that's related to research design. We'll review steps in the research cycle and then focus down on the step of formulating hypotheses, looking especially at the advantages of trying to have multiple hypotheses. Part 2, after beginning with a discussion of the advantages of constructing formal experimental designs, will focus on the basic concepts of experimental design, including object protocols, measurement protocols, and treatment protocols. And after part 1, I'll take a few minutes of questions, if there are any, and also again after part 2. And then parts 3 and 4 will be done together with questions at the end. Part 3 will look at the more common type of experiment that we see in preservation and conservation research, the multiple object design. We'll talk about using a design check sheet and research design flow charts or sketches or diagrams. And then we'll look at some case studies of what I think are really good multiple group designs. In part 4, we'll move into single object studies and a few other design variations. We'll look at some case studies of how statistically valid studies can be conducted when we only have a single object available. And then we'll conclude with a discussion of screening experiments and treatment trials and how these might be useful in conservation. So we'll get right into the meat of things now with a brief overview of the scientific method as an approach to research. There was a classic paper that was published in 1964 in the journal Science, and it received a lot of attention at the time it came out and for years afterwards. It's still a classic and it's definitely worth reading, although it's now dated, especially you'll notice in sort of genderized language that's used. But the main point is that he noted that some fields of science seem to advance at a much faster pace than others, and he wanted to try to understand why. He proposed that the main difference is really an intellectual one, that rapidly progressing fields systematically use an approach that he called strong inference. He says that includes steps used by all fields of science, but in some fields these are systematically and regularly applied as the standard accepted approach. And this method includes, first of all, devising alternative hypotheses, then devising a crucial experiment or a series of them with alternative possible outcomes, each of which will as much as possible exclude one or more of the hypotheses. Then carrying out the experiment so as to get a clean result, and recycling the procedure to develop sub-hypotheses or sequential ones to try to refine the possibilities that remain, and doing all of this as part of long-term research programs. He notes that while this is understood to be the way science works, actually in practice we often forget to clearly follow all of these steps. Instead, what happens is when the paper is written at the end of a project, it's written as if this process had been followed after the fact. Or he says we find ourselves doing what's more like busy work, doing analyses without appropriately setting up good hypotheses, maybe being too method-oriented rather than problem-oriented. And in contrast, in some fields, he felt that regular and explicit use of alternative hypotheses with clear exclusions is the norm, and that leads to fast and clear progress. He discusses in detail why just affirming a hypothesis is insufficient, because there might be a lot of factors or explanations involved that we haven't really thought about to test for. That science really advances by disproving until all of the exclusions have been exhausted. And then the possibilities that remain are really more strongly supported. Two examples he cites are high energy physics and molecular biology. He said that anytime you walked through Francis Crick's laboratory, for example, you would see blackboards where the hot new results from his or some other lab were written, and then below that would be 2 or 3 alternative explanations that people have written down, and then others would fill in with a series of suggested experiments that could reduce the number of possibilities. Platt talked about how Enrico Fermi, the physicist of the early to mid 20th century, who developed the first nuclear reactor, contributed to quantum theory, nuclear and particle physics, statistical mechanics, and was a Nobel Prize-winning scientist renowned for both theory and experiment, so clearly a successful scientist, that he explicitly and regularly practiced this method of spending time every day thinking about and writing down his thoughts, including writing down possible hypotheses and possible tests, and using this as an expansion of a more typical notebook that you would use for laboratory work. Platt called the strong inference method the logical tree. So if you have multiple hypotheses or explanations for what you've observed, and you design an experiment that should exclude one of those possibilities, then it's like a tree. At the first fork of the road, the experiment sends you to one branch or it closes off another. And that eventually this leads to forward progress rather than going around in circles or not going anywhere at all. We're familiar with these sorts of logical trees because they're used as identification keys for things like plants, trees, pollen, or here this example is for identification of parasites that can infect animals. And we use these in conservation. For example, for pigment identification keys using the results of polarized light microscopy or simple mechanical tests. And so as you go through this tree, certain tests will allow you to exclude a possibility, and eventually it takes you down to where you can pretty confidently identify what pigment you have. Another example of really thinking carefully through your experiments that Platt talks about is Louis Pasteur. This was a French chemist and microbiologist of the late 19th century, and he made remarkable breakthroughs on the causes and prevention of diseases. He developed the first vaccines for rabies and anthrax, did experiments that supported the germ theory of disease, developed the pasteurization method of milk and wine to prevent disease, and he also made breakthroughs in understanding of crystal structures in chemistry. So Platt noted that every few years, Pasteur moved into studying a new problem where there were already a lot of experts who devoted their lives, or at least many years, and then he would solve problems that no one had been able to do before, which I'm sure was very annoying to some of those people. Though clearly it wasn't encyclopedic knowledge that was the secret, nor was it amount of time spent doing the research. Platt said it was Pasteur's systematic application of strong inference, and he contrasts this approach with general surveys and observational studies that often don't have clear alternative hypotheses or tests that could exclude a possibility, but which are designed really just to gather data. And he says we call this science and say it's important because it's collecting information that might be useful later as another brick in the wall of science. But Platt said that most of those bricks just basically end up lying around in the brickyard. If we're just collecting them in the hopes that someday someone else can use them to answer a question of interest or address a hypothesis. A good suggestion that comes out of following this method is the idea of keeping a notebook, whether it's physical or electronic or on smartphone notes. That goes beyond the idea of just keeping the laboratory notebook for recording our actual experiments and data, but it's used for constantly reflecting and writing down new ideas, new observations, hypotheses that might come from those, implications of those hypotheses, and therefore how you might design experiments to test them, and any other brainstorming ideas that come from just sitting and spending at least a few minutes every day thinking about the problems that you're interested in working. So these are the basic parts of the scientific method that we'll now discuss individually in a bit more depth. The observations, specifying a research question, constructing hypotheses, developing the implications or inferences of those hypotheses which lead to the design of experiments, explaining the rationale and the logic behind our hypotheses and our implications, conducting experiments, doing the analysis, publishing the results, and then building on them to create a long-term research program. So first is, when you observe something intriguing, puzzling, or problematic, write it down. If a literature search doesn't show that the answer is already known, then this observation could be the basis for a fruitful research project, either now or later. Next, based on that observation, specify or formulate the exact question under study. And you're going to want to choose a research problem or question that's clear and answerable within the period of time that you have available to you. And that's really important. So if you have 1 year to carry out a research project, make sure that the research question, to the best of your ability, is something that could be answered in that period of time. You'll have a very different type of research question if you're going to conduct a 3-year project. And if you have only 3 months, it will be different altogether. There are different types and levels of research questions. Clinical questions might include something like, which coatings in this group will pass or fail various tests for use on outdoor bronze sculptures? We'll look at what phenomena are present, what processes are occurring, what results do various treatments have. So these questions may give you information that you need to conduct further research, and they're always needed as a first stage in a new scientific research program. Other examples of clinical questions could be, does a particular colorant fade over time? If so, how fast? What factors might affect the fading of this colorant? Is it light, exposure, temperature, humidity, air pollution? Maybe which of 3 proposed controlled environmental parameters most inhibit the fading of this colorant? Scientific questions might be something more like, why do some coatings hold up better in this particular environment over time? What do they have in common? With these types of questions, we can then predict future coating performance better. We can gain knowledge about the underlying reasons for the observed behavior, not just observed behavior. For example, if we try to answer the question, why are some silver coatings permeable to hydrogen sulfide, so the silver turns black, while others aren't, we can then use that information to select appropriate coatings for future testing. So here we might be looking at things such as, why are the observed phenomena present? Why do certain processes occur? Why do various treatments produce the results observed? It builds on the knowledge that we've gained from answering those clinical questions, but it allows you to go further. It allows you to predict the outcome for conditions that you haven't yet tested. So we would look at, why does this particular colorant fade over time? If ozone, for example, accelerates the fading of the colorant, what is a mechanism of that effect? Knowing that mechanism, can we then predict what other factors might also cause this colorant to fade? And can we predict what will happen in the environment of a particular building? The difference really is that of product testing versus scientific research. If we can better predict which coating should work well, we can do more efficient selection of coatings for testing, or even design an ideal sort of coating. So with a clinical question, we might rank several coatings or colorants by the degree of change in a similar environment over time. And then the scientific questions would try to look at explaining the ranking and identifying the underlying relationships so we can predict what change might occur on untested products. But we need both of these sorts of questions. There are other kinds of questions we might ask also. For example, how much variability do we see in results? And is the change, such as fading, that we're seeing significant enough to be worth worrying about? Once a research question or problem is developed, then the next steps in research continue with developing your hypotheses. So these are going to be the possible answers to your research question or possible explanations of your original observation. Platt referred to a paper that had been published in the journal Science way back in 1890 by T.C. Chamberlain. And then after Platt's article came out, it was reprinted by Science again in 1965. Well, again, of course, it's dated in language, even more so than Platt's. This paper does make a few interesting points about the usefulness of trying to have multiple hypotheses rather than a single one, and how that can encourage creativity. He talked about an argument that was raging during his time regarding the origin of the Great Lakes Basin. There were 3 competing theories that were being sort of violently argued. One was that they were river valleys whose outlets were blocked by glacial debris. The second was that they were excavated by ice, and the third was that they were made by crustal deformation. And it turned out that it wasn't actually a case of which one is supported, because none of them could be refuted. But the real question ended up being, how much does each contribute to the explanation? So often coming up with a single hypothesis may be too limiting, because a problem often has multiple facets that can be better identified if you're considering multiple hypotheses. Chamberlain also talked about how if you have only one hypothesis, it can become somewhat like what he called an intellectual child. If it's our only one, we may simply not see anything that doesn't support it. We become invested in trying to find things that support it, and we just don't see things that might refute it. Now, of course, there are many journals that won't accept papers that only report on lack of hypothesis support. So it's also sort of a scientific survival instinct to try to support a single hypothesis. Chamberlain put this a lot more poetically, so I'm going to read the sentence from his paper on this. So, quote, "The mind lingers with pleasure upon the facts that fall happily into the embrace of the theory, and feels a natural coldness toward those that seem refractory. Instinctively, there's a special searching out of phenomena that support it, for the mind is led by its desires. There springs up also an unconscious pressing of the theory to make it fit the facts, and a pressing of the facts to make them fit the theory." Unquote. So while one hypothesis might immediately come to mind when you're looking for an explanation for a particular observation, if we can develop a habit of then trying to be more creative and come up with additional ones, that can feed creativity, and it might ultimately provide for a more robust explanation. But it also means that we'll have less invested in there being a specific outcome, because if the original hypothesis is shown to be refuted, then one or more others still might emerge as good possibilities. So we're still going to have a good result. And it's good practice to try to bring into view then every rational explanation for our observation, and try to develop every possible reasonable hypothesis we can think of, not just the one or two most obvious ones. We'll look first at a clinical example. Here the observation is that some adhesives used in conservation of a particular type of paper discolor. And the research question is, which of three adhesives will discolor the least on this particular paper type? Now, in trying to develop alternative hypotheses, you can always negate the first hypothesis to come up with a second, if you're truly willing to take both outcomes seriously. And this means that no matter what the experimental outcome, you're still going to have a result. Something's going to be supported. You won't be able to you should at least hopefully be able to clearly eliminate one of them. And here we have as our two hypotheses, first, that there's no significant difference in discoloration over time for these three adhesives on this particular paper. And the second hypothesis is the three adhesives are significantly different enough to be ranked from better to worse. So clearly one of these will get refuted or eliminated. And looking at a conservation example, one of my favorite ones is from a paper that Helen Alton, a conservator, wrote and presented back at the very first materials issues in Art and Archaeology Symposium. And it was an evening session running late at night, I think 7 to 10. And most of the other presenters, if not all, were scientists, glass scientists, and Helen was the only conservator that I recall. And it being late at night, and the other papers were very convoluted and hard to follow. People were nodding off, falling asleep. With Helen's paper, everybody woke up, paid attention. It was clear and easy for anyone to follow, whether you were an expert in that subject matter or not. And she was looking at glass from a 13th to 14th century archaeological site. Her observations were at this wet archaeological site. There was a lot of variation in the amount of visible deterioration that could be seen with different wet glass fragments. The second observation was that air drying the glass often seemed to cause loss of translucency, a visual emphasis of defects, or even cracking and fragmentation. So her research questions were, for both observations, what is the cause? She developed 5 multiple hypotheses, and all of these had rationales that explained why she was proposing these hypotheses. So the first hypothesis was that waterlogged glass is damaged by air drying. And her rationale is that she found published sources that indicated air drying might damage glass by causing loss of material. The second hypothesis was damage by air drying is more severe in the more corroded samples. And her rationale is that the more corroded samples might be damaged more severely by air drying through loss of structural strength. Third, the physical removal of water causes damage to the glass. And here her rationale was that water might act as a bulking agent, consolidating part of the glass structure, or surface tension of the water might cause physical damage to the glass during removal. The fourth hypothesis is that the refractive index of the water merely masks damage already present. And the rationale is that the removal of the water itself is not perhaps damaging the glass. It's just that the damage that was already there is no longer being masked. And fifth, she hypothesized that maybe initially different glass compositions were causing differing glass corrosion. And the rationale is that the different compositions may behave differently on water removal, and so will have different initial corrosion appearances. She then went on to present tests for each of these hypotheses, and she again gave good rationale for why those tests were good to address these specific hypotheses. So I invite you to go and look at that paper, and it's in the list of references up on the NCPTT website. The next step is to explain all your relationships and rationales clearly, as Helen Alton explained why her multiple hypotheses each might have validity, and therefore we understand that they were worth examining. And this is an important step because it's not always clear to others what your reasoning is. And it might not even be clear to you later on, or if you step away from the project for a while. So this type of lack of clarity, where the rationale or reasoning isn't crystal clear, is also often a reason for papers reporting on the work to be rejected by journals, or for grant proposals to be unsuccessful, because the readers or reviewers will look at that and say, I don't understand why you think these hypotheses are answering your question, or why these tests were being done, how are they answering your, addressing your hypotheses. The next step is to infer, and this is crucial because you're going to be basing your experiments on these inferences. What are the implications of your hypothesis? If it's true, or if it's going to be refuted, what do you expect to find? And you need to be clear here in order to set up tests that are going to clearly and logically connect back to your hypotheses. So it's clear to everyone that your experiments are in fact clearly and cleanly eliminating or testing your hypotheses. So if these combinations of hypotheses and their implications, they need to be explicit and written down in order to perform clean experiments and get a clear result. If you don't have any hypotheses and you're just collecting data that you're then going to use later to try to come up with some interpretations or explanations, well, you're never going to be wrong, but that type of work also tends to make really slow progress, or even no progress. And funding and publication is also likely to be more difficult because reviewers want clear hypotheses and test implications. One example that I know is I had a colleague who had a fellow researcher die, unfortunately, and he left a huge amount of data that wasn't accompanied by any written hypotheses or test implications. So the lab team needed to do something with this to get back to the funding agency. They worked for a long time just trying to dredge through the data, to do various analyses to look for significant associations, to try to come up with some useful result. They did finally produce a tiny, not very important result, but it certainly wasn't worth the years of data collection and the person hours that were put into that. So a hypothesis also should not be so weak that no reasonable testing program could ever disprove it, such as this treatment might be valuable under some circumstances. This isn't really the way to advance the field or to make efficient use of research time and funds. You also don't want to try to say everything in one hypothesis and overcomplicate it. But you do want to try to say something worthwhile. Hypotheses should not be value judgments outside of the realm of testing, and this is something that's especially problematic in social sciences. But clear hypotheses with rationales for why those are possible explanations for your observations and clear test implications means that others can follow your thinking patterns and you yourself can follow them even years later. We'll look at a couple of hypothesis examples for scientific research questions. And here we have an observation that some adhesives used with this particular type of paper discolor. The question is, what factors determine how much an adhesive will discolor on this paper? The first hypothesis might be adhesives with certain chemical bonds susceptible to hydration will react with water, causing discoloration. And the implication here is that color measurement of some adhesives subjected to varying humidity will show greater discoloration after exposure to high humidity. The rationale is that excess moisture allows hydration reactions to occur. And a second hypothesis might be adhesives containing a particular impurity will discolor over time. The rationale might be that this particular impurity is highly reactive and it's known to be something that can be introduced during adhesive synthesis or processing. An implication might be that color measurements made before and after artificial aging on adhesives with and without the impurity will show greater discoloration on the ones with the impurity. So here we can see that both hypotheses could be supported rather than negated by experimental work. And then we might be looking at what degree to each of these contribute and get maybe a better explanation for what to expect. And the next step is to design a testing program using those test implications we've just come up with. So think back to that idea of a logical tree and use that as sort of a roadmap for the design process. And this type of design plan, where you're going to at least make an effort to eliminate at least one of your hypotheses, will allow for some real conclusions, and that will then strongly support whatever remains after your attempts at elimination. Simplify, simplify, simplify. And this is actually quite hard to do because we all have a tendency to try to design one experiment that can test for all possible variables. In conservation and preservation, you'll be constantly pressured by colleagues to add this or that variable into the test. But don't do it. Focus on trying to exclude at least one of your possibilities in a simple test that will give a clear result for that, and then move on to the next thing to test for. For example, if you're mainly interested in how well a coating for silver can protect against hydrogen sulfide, then the first logical test would be to test your potential coatings on coupons of 1 silver fineness under exposure to just hydrogen sulfide. If a colleague notes that maybe application method also has an effect. So you should test 3 common application methods. And maybe there's a difference between fine and sterling silver, so add in that variable. And maybe we should also check on how the coatings perform on copper and lead too. And maybe metals of different geometries will vary. And so maybe we should try some curved coupons and some flat coupons. And what about trying to vary the coating thickness too? Pretty soon you're going to have an overly complicated experiment that will take much too long to do and is likely to give a result that's going to be difficult to interpret. So try not to do this. You can instead do a series of experiments in progressive logical steps. You don't need to do it all in one highly complex long experiment that may not have results you can clearly interpret or explain. So what is the simplest experiment that can exclude one of your alternatives? This is called a crucial experiment. And here we want to use the thinking again to come up with ways to try to exclude one or more hypotheses in the simplest, shortest, most clear experiment you can versus the longest and most complicated and most expensive one you can. So we carry out the experiment. Analyze the results. And this may require statistical analysis, it may not. But however you're going to interpret your results, what you're going to be doing is looking at those actual results and comparing them back to those test implications you created for each hypothesis. The next step is to publish your results, so we don't have to all keep reinventing the wheel, and then make sure to do a thorough literature search at the outset when developing your research questions and hypotheses so that you're not reinventing the wheel. This is actually another reason for papers often getting turned down for publication, because we might sometimes see scientists in other fields who didn't check the conservation science or conservation science literature and are not aware of the work that's already been done, or conservators or conservation scientists who didn't check the allied literature, and that will get often caught in peer review. Next step is to build on your results if possible. Unfortunately, this is most often dependent on long-term or follow-up funding, and that's often difficult to find in our field. So publishing then is even more important, because someday someone else may get funding to build on your results, or you may have to wait a couple of years before you can continue with that line of research. So at least publishing the initial results is going to get that information out into the field in the meanwhile. And this is a drawing of the research cycle, which you can see is not linear. You often need to backtrack and rethink things again when something doesn't work out the way you planned. And my experience is that always happens. Something somewhere always goes wrong or is unexpected, and it means you're then going to have to start over and tweak your design. So you may start up here with your observations, you develop some hypotheses, you create a design, and you start to do your experiment, and then you find out that it's not going to work. You have to go back and either redo your design or reformulate your hypotheses. You finally run your experiment as you're analyzing things. Maybe you didn't get a clean answer and you decide you had something fatally wrong in your design, or even back up with your hypotheses. Finally, you get through here, you publish it, and then you're going to create sub-hypotheses or sequential hypotheses to hopefully continue on the research. And I'll just show one quick example of where things go wrong and research is not linear. I was working on a silver coatings project with a rather large team, and things got a bit overcomplicated. After weeks of planning as a team for which coatings to test, on which shapes of the coupons, which application methods, how to set up the accelerated aging chamber, which pollutants to introduce, and when and how to monitor them, etc. We finally got started on the actual experiments, and we got the coupons in the chamber and began what was supposed to be a long-term test. But it turned out that one of our primary coatings that we were testing, and we were testing it because it was recommended and used in the preservation of silver, turned out to be completely permeable to hydrogen sulfide, at least at the thickness that we had applied it. And that's a common off-gassing product. So within the first week of testing, all of the coupons with that turned completely black, and it would then swamp out the results of any further analyses we wanted to do. And so we realized we had to stop and rethink the entire experiment. And later we actually found out that the same thing had happened to some silver collections where this coating had been used, but no one had ever reported that in the literature. So even when you try to think about every possible aspect of your design and you've done a literature search, things can still go wrong. But think about how many more things could go wrong if you didn't spend a lot of time thinking, planning, writing out rationales, and checking the literature. So I want to end this section by quickly reviewing some of the advantages of trying to come up with multiple hypotheses prior to undertaking research, rather than just grabbing onto and focusing on the one that seems most obvious to you. It can encourage creativity. You may come up with some potential answers that had never occurred to you before. And it can also encourage consideration of more complex causes of a problem. But you may have multiple solutions that are all contributing. So you may end up with a more sophisticated answer. So you try to think of as many potential answers to your research question as possible and not just stop at one or a few. I invite you to practice this at your leisure with a hypothesis brainstorming practice vignette that's been posted on the NCPTT website. And there's a couple of brainstorming exercises there where you can try to think of as many hypotheses as possible that could be possible answers as to why deterioration is occurring in two particular case studies. And you should be able to come up with 8 or 10 hypotheses. Some of them are things that you might be able to quickly eliminate through a literature search. Others could be eliminated through some simple tests, and others may require more detailed research. And now we're going to pause for a couple of questions, if there are any.